Hi, cellular respiration might seem hard at first, but when you break it up into its individual stages, it's more digestible. This is what my partner and I will try to do. Cellular respiration is made up of three metabolic pathways. A extra pathway will follow when no oxygen is present, but we will talk about that later on. The first pathway is glycolysis. This takes place in the cell's cytoplasm. Its job is to break down glucose into pyruvate. It does this in two stages. First is the energy investment stage, where two ATP are used to prime the glucose molecule. Then it is cleaved into two molecules of G3P. Then comes the energy payoff stage, where the two G3P are first oxidized by NAD+, which will be reduced into NADH, a high electron carrier. These will be important later on, so try to keep a close eye on them. Then we will make some ATP through two substrate phosphorylations. Since all this happens twice, once for each of the G3P, we will make two NADHs, four ATP, and a net of two ATPs, taking into account the two that were first used. Most importantly, we are left with two pyruvates that will be used in the next pathway. After glycolysis is where one or two things may happen. If oxygen is present, the pyruvate will follow anaerobic respiration. In other words, fermentation. In humans, we perform lactic fermentation. The pyruvate will be reduced to recover the NAD plus used up in glycolysis, in turn making lactic acid. This is actually what makes your muscle sore. Fermentation, however, is not very efficient. This is why the second pathway is preferred. Now, if oxygen is present, the pyruvate will travel into the mitochondrial matrix to enter the Krebs cycle. Before it enters the Krebs cycle, the pyruvate must be oxidized in this step. In this step, another NADH will be produced alongside CO2 as a byproduct. What you are left with now are two acetyl-CoAs, one for each pyruvate.
Now we're going to be talking about the Krebs cycle. Acetyl-CoA will enter the Krebs cycle by binding to a 4-carbon oxaloacetate, which will result in a 6-carbon citrate. The citrate will be oxidized twice by NAD+, resulting in 2 NADH and 2 CO2s. Now the resulting 4-carbon molecule remains. This molecule will undergo substrate phosphorylation once, producing ATP. Then it will be oxidized twice, once again, this time first by FAD+, just another high electron carrier, and NAD+, producing FADH2 and NADH. What is left is the original 4-carbon oxaloacetate that will begin the cycle again. This whole thing happens twice per glucose molecule, once for every acetyl-CoA. Now that you are done with the Krebs cycle, comes the part where most of the energy, ATP, will be generated. All those high electron carriers that were produced in glycolysis and the Krebs cycle will be used up here in the electron transport chain. The electron transport chain is made up of 3 proton pumps and one very important enzyme called ATP synthase. They are located on the inner membrane of the mitochondria. As the electrons are dropped off by NADH and FADH2, they will travel down the chain. Each time they will pass through a pump. Their energy will be used to pump a hydrogen into the intermembrane space. The final low energy electron will be dumped onto oxygen generating water. This will set a chemiosmotic gradient in where the concentration of hydrogens will be higher in the intermembrane space than the matrix. This will lead to hydrogens wanting to diffuse back into the matrix, and they will do so through ATP synthase. This enzyme will take the hydrogens and use them to produce ATP, about 1 ATP for every 4 hydrogens. This is the end of cellular respiration. In total, 30 ATPs were produced, 6 CO2s, and 12 H2Os will be produced. I hope this better helps your understanding of cellular respiration. Thank you.